All right, we have another view of the bottom here. We are at, oh, I just changed that. We're at about 1,829 meters, about 6 meters above bottom right now. Looks like there's a lot of sponge on the bottom here.
You already took the an eDNA sample at the bottom, right?
No, we decided we'd wait until we reacquired bottom up here, and we'll take the eDNA sample shortly.
That's great. Pick up lots of sponge.
Exactly. I think I see a black coral also, several black corals.
Almost as interesting.
Yeah, this looks like it would have been a good bottom site too.
Yeah, very much so. That looks like a nice glass sponge, that white guy there to the
It
left.
did fire, right? Yeah, I think I see that second one's closed. Yeah. All right, so we got that second collection, but I have to go back and redo the paperwork here. Second, this. Okay, we're set there. We now have our second water sample for eDNA. We took one in the deep scattering layer on the way down, and we have the second one. And we're just having a little look around here, what's on the bottom. See lots of sponges and black corals, light dusting of sediment on an encrusted bottom. Pilot's watch lead. Watch lead 2, pardon me. Can you zoom in while we're sitting here? Yeah, let's zoom in on the just what's ahead of us. Yeah, great. So very nice. Looks like a glass sponge to me. Okay, that's good. We can come back and then look at the left side. There's a yellow one.
Looks like a uretid.
Oh, uretid. Thanks, Alan. Looks like kind of an encrusting sponge there, growing on another sponge, the yellow growing on a white one. And then, yeah, just below it, Those look like demo sponges just above the laser there. Maybe a little cup coral. And I see several Stichopathes black corals, the unbranched corals. That's great. Okay, we can pull out from there. I see a Galatheid crab tucked in the rock. And then over to the left a little bit more, I see an urchin, another sponge. And then, yeah, that's what I was looking for. That looks like, that's a Zoanthid. The yellow thing on the lower corner, the Zoanthid is overgrowing another colony. And what's on there? Is that a Pycnogonid? I think it is. Yeah, it's pretty dark. Yep, that's Pycnogonid, and there's also a brittle star, snake star, basket star, excuse me, wrapped around the branches of what looks like a plexaurid of some kind. And there's another cup coral. So there you go. Oh, and there's one sea star. So now we have a little look at what's on the bottom here of this feature, and we're going to get ready. Yeah, go ahead. We can get ready to do our midwater trawls.
Not a trawl.
Sorry, thank you. Transects.
Like that word.
It started with a T. Yeah. It's still kind of early rolling, but I appreciate that. Great. So it looks like we have a fairly productive and perhaps diverse bottom community on this local topographic high. And pilots watch lead too. I'm not sure who it's appropriate to ask, but once we're set up, if you could give us at some point a sense of the currents that you're experiencing, that'd be helpful. Thank you. Okay, folks on shore, so we're 15 meters off the bottom. We're pretty much set up. We're facing south across the plateau of this feature. And for those of you on the phone, Adrian and Mike and Alan, you know, I'm happy to stay quiet while you guys do the call-outs and the narration of what we see.
Alan's really good at that.
You made me laugh.
Will we get a verbal notice when the transect is officially started?
Yes, you will. I'll let you know when it's started. Right now, what they're doing is they're checking out what are the currents. I heard the pilot say it was moving port to starboard, and so he's orienting the ROV such that they can drift and get basically the slowest rate that the plankton will be approaching so that we can get the best zooms. So once they've figured out what that orientation is, then we'll start to transect.
Great, thanks.
Hey, Mike, I don't know if there's anything you wanted to say about why we're targeting the benthic boundary layer. This is just, it's a really interesting environment because this is where the marine snow is accumulating from above. They have all the benthic diversity and then lots of the pelagic organisms. There seems to be a higher diversity and abundance of pelagic organisms really close to the bottom here to take advantage of this sort of like more nutrients and more diversity around.
Yeah, probably a lot of viewers are not familiar with the term benthic boundary layer, but that's the layer of open water that's closest to the bottom.
All right, good morning, science. This is RV Navigator. Just letting everybody know the pilots are set up, and we are about to begin our first midwater transect. Depth is currently 1817 for the vehicle. And as soon as we get to the top of this next minute, we will begin our first deep water transect this morning.
Copy that, Navigator. Thank you. I think you all heard that on shore. On the top of the minute, we will, 4 seconds, start our first transect. Transect beginning.
Okay, I'm on OKX now with you,
Okay, just
Scott.
to finish what I was saying about the benthic boundary layer, the interaction between current flowing across the bottom stirs up the bottom, but also it's an important area because the animals in the water, like that heating gnat that we're looking at right now, are vertical migrators, many of them, and when they migrate down to the bottom part of their vertical range, it intersects with the bottom, so that concentrates pelagic animals, and also there's a lot of benthic animals that swim up off of the bottom. So it's a really interesting layer in the water column, and it can, depending on how strong the current is and the topography of the bottom, it can extend for 100 meters or more above the bottom. See if Alan wants to say what this is. It looks like a ctenophore.
No, I think we've got like a little dipheid swimming bell from a siphonophore.
Yeah, that's what I thought it was, Alan. Just for...
I saw some of those reflecting there.
There looks like a Keating-Nath, an arrow. No, that's a fish, actually. Looks like a myctophid, midwater fish.
Yeah, it's a cyclophony. That's the
White
most
one.
abundant vertebrate on the planet. It's the genus cyclophony. They're small fishes. They look a lot like Keating-Naths, and they're easy to confuse with Keating. But they're fishes, and there's so many of them in the deep environment of the deep midwater that if you do the math, you find that there's more of them than any other kind of vertebrate.
So, Alan, I think that looked like the swimming
Now
bells of siphonophore. Yeah, go ahead.
I'm just saying that we got another good look at the dipheid siphonophore tuna feet.
And at the same time, there was something red there that was an isopod.
Yeah. It's a good look at a cyclophony there. They can be extremely abundant, but even when they don't appear to be abundant, the volume of the deep water column is so great that there's huge numbers of them, even if they're all bunched up together.
Okay, it's going up too fast.
That looks a little bit like the ropalonematid Panthochogon. Yes, Google confirms Panthochogon hecali.
That was the small jellyfish, Alan?
Yeah, the small jellyfish, little sort of hemispherical shaped.
Yep.
Comes in a couple different color morphs, but a lot of them are very orange, like that one appears.
And is that a hydromedusa or a scyphomedusa?
That's a hydromedusa in the order Trachea Medusae.
Trachea Medusae, thank you. So, yeah, just if I have time, just to translate for the general public. So, there's lots of things that get called jellyfish, and they're not necessarily closely related. So in the case that they were just talking about there, that's something that's related to the siphonophores and the benthic hydroids.
Yeah, and it's really interesting. So many of the jellyfish in their life cycle, they have a polyp stage, and the group of called Trachea Medusae, they're all direct developers. So they sort of skip that polyp phase and develop directly into the medusa.
Is this an arrowworm, Keating-Nath? Or is that a fish?
Yep.
I think that's neither.
Oh,
that's good.
Like
we have
both,
three opinions.
But I don't think it's either one.
Well, unfortunately, it drifted by pretty quickly. I'm looking at video replay, and I can't quite tell.
Dougal said that it looked dead or infected. An infected geograph in the chat.
Well, that's an interesting idea. Yeah, so this looks like one of those same siphonophores we saw earlier. I think the suggested name was Cheniphes. Is that correct?
Cheniphes.
Cheniphes, thank you. And what you can see is there are these little swimming bells. You see there, I think I count 3 of them maybe, and each of those will contract to produce a little jet propulsion. So essentially a siphonophore, or more common would be the Portuguese man-o-war that you see up at the surface. This would be a subsurface deep-sea form. Something just left of center. Looks like a discarded larvation house, perhaps. Oh, maybe not discarded. I think I see the tail beating in there. Yeah.
Still in there.
Yep, the larvation is still in there. So this is a pelagic tunicate. The small invertebrate that builds a mucus house that it uses to filter all this marine snow, these organic particles that you can see drifting about here. Pretty fascinating animals.
Another important thing about larvaceans is that they are really important in the carbon cycle on the planet because this big mucus house accumulates stuff on it, and then eventually it gets so clogged up that the animal abandons it to make a new one. But the abandoned larvacine house sinks down to the bottom, carrying a whole bunch of carbon with it. And because it's so big, it winds up transporting it very fast down into deep water.
Absolutely. And here's another one. You can see that tail, that shimmering appearance is the tail that's beating, and then the more concentrated white part that you see on the right there is where the main part of the body is, and then everything else that you see around that is this gelatinous mucus house that Mike was referring to that concentrates carbon, this marine snow. Oh, this looks like a worm, perhaps, a polychaete. Oh, look at that. This would be nice to get a good view of.
Yeah, that would be really nice.
Yeah. So is it a polychaete or is this some kind of crustacean? I'm not sure what that long tail-like thing is. It must be eggs.
Oh, no, I think we're seeing a horned ctenophore. This is
oh, you're right.
That's yeah, we definitely want good footage of this.
That's fantastic. I've never seen a horned ctenophore. So those are the retractable tentacles, right? And I can see the little branchlets on those tentacles on the upper one.
Yeah. So it has these 2 large tentacles that it deploys, and then each of those should have side tentacles off of it, very fine ones, it looks like in this case.
Yeah.
And then this particular...
I hate to interrupt, but if we can collect, Dougal wants a collection.
It's already, it's gone. Sorry.
I thought so. Sorry about that.
No problem.
Ouch.
We were trying to get good imagery of it. This looks like a mysid opossum shrimp to me. Oh, you can look on it.
So shoreside, definitely we'll need sort of a conceptual idea of what you guys want to sample ahead of time because we don't have a lot of time as we're coming up on it before the vehicle passes Sirius.
if we see one again, we will certainly make an attempt to get it.
Understood. Thanks, Roland.
Yeah, one problem is that we don't know what we're going to see in advance. So it's hard to tell you what we want to sample.
Yeah. Well, I think it's pretty clear that that was really interesting. So if we are lucky enough to see another one, we'll do our best to sample it.
That would be great.
This looks like a Ketignath, an arrowworm. It's head down, muscular tail up. Yep.
I think we're seeing the gonads there towards the tail end.
Mm-hmm. There's lots of Ketignaths in the water column. They're sit-and-wait predators. You could see that one was just kind of idling head down, but they can move forward pretty quickly, and they have these pretty ferocious-looking grasping spines at the head end that they use to capture their prey.
I think we were seeing both their eggs and their sperm. They're hermaphrodites, so we saw both pairs of gonads in the back end of there.
Yep.
You see how close we are to the bottom there.
Yeah, it's not something I'm used to seeing on these transects. So right ahead, just to the left, upper, yep. It's another cyclothony, one of the midwater fish, I believe. Get a confirmation, and then there's something to its right. I can still see it there. It's slightly, it's like at 1 o'clock from what you just zoomed in on, not far at all. There you go. Looks like maybe a copepod or a shrimp of some kind. There we go. It's definitely not a copepod.
Yeah, I think it's a shrimp of some kind.
Yeah, I couldn't tell, Mike, if it was some kind of Caridian or if it was, I don't think it was a mysid.
Is it a surgested? Pretty long.
Yeah, it's probably it's probably the other group, not the Caridians, but there's a new name for them that escapes me right now.
Those dendrobranchiates? Dougal Lindsay is suggesting perhaps a Eucopia mysid.
And we saw those little pair of balls. Dougal says those are Phaidarians. These are unicellular organisms that used to be classified with radiolarians, but are a separate group.
Oh, that's cool. Those are really quite large. There's something white just to the right of center. Another Ketignath, I believe. Yep. One of the arrowworms again. This one looks different. The gonads are larger, or it's just a further stage of development. You can see that they can move pretty quickly when they want to. Yep.
Run away!
Exactly. There's some, I'm not sure how it's moving pretty quick across the screen. I guess we're reorienting to have it coming into us. Yeah. Not sure if it's a mucus string. Looks like a, is that like a pseudomorph, Mike, you think?
Yeah, it just looks like mucus to me. I don't think it's any. So I don't think it's a pseudomorph. I think it's just some marine snow.
There's 2 more balls on the, oh, they just separated. Never mind. Thought it might be the Phaedarians again. Let's see. Kind of looks like mucus again. That might be something. No. Here, though, just in the center, coming from, go ahead. Yep, go for it. There's a shrimp for sure. Much larger.
Yeah, that's a Caridian.
Very nice. You can see that red color that a lot of these deep water plankton have, zooplankton, and the that red wavelength doesn't travel very far, so they're essentially invisible. Spheronectes, Dougal suggests, as a genus name for that shrimp.
I think he's suggesting that it can't thyra.
It can?
Spheronectes is a siphonophore.
Oh, I'm sorry. Okay.
Yeah,
Thank you.
The shrimp, I think, was a Canthophira.
A Canthophira. Very good. Sorry about that. It's the delay, right? I'm not familiar with a lot of these genus names.
All the way around the world.
Yeah. It was pretty remarkable to be getting these IDs from Japan right now. Let's go left. Yeah. So
another cyclothony.
Yeah, so this is a large larvation house. Again, you can see the beating of the tail of the larvation in the upper center, and below it you see a lot of the filtering units of that mucus house. It's a remarkably complex construction, that mucus ball that they use to trap all this marine snow.
Yeah, it might be worth mentioning that what you can see is actually the inner filter. There's also an outer ball of mucus, which can be quite large, actually, for some of these things. And it's really difficult to see on the video. So in addition to that inner filter, there's a lot more mucus involved.
Yeah, think about like a baseball inside of a basketball. There's a little copepod, just one off upper right. There we go. It's in the center now. It's probably so small. Drifting down. Yep. They're hard enough to identify when you're looking at them under a microscope. What about the upper center? See what's going on there.
Thinker?
Yeah, it's a mucus strand of some sort. It might be a detached tentacle from something.
Yeah, oh, I see. Okay.
From one of the gelatinous. Sometimes when they get startled or disturbed, they can drop their tentacles and swim away.
They're deciduous. Dougal Lindsay's noting that you can sometimes identify those larvations to the family and sometimes to the genus, which is a more specific level, just by the structure of that inner house and the filters. So let's see, I think that's maybe a Ketignath going right, and left was some kind of shrimp. Yep, that's a Ketignath. Well, there's something on it, huh? Yeah.
Yeah, maybe a parasite.
Yeah. There's definitely something riding that Ketignath. Interesting. That's the first 20 minutes of the transect. 25 minutes to go. Another Ketignath, arrowworm. Looks like maybe another larvation house there if you want to just zoom a little more. I do believe this the structure of this one looks different to me.
I was just going to mention another interesting thing about the larvacean houses is that they have also been shown to pick up microplastics, and so they're, in addition to moving lots of carbon to the sediment surface, they're also moving plastics out of the water column to the bottom.
That's really interesting, Alan. Thank you. There's something white just coming into the center. Slender. And, of course, it's another arrowworm. Different individual, no parasite on this one. Let's find another one of those ctenophores. That was a little bit bigger.
Yes, please.
That's probably another cyclothone, I think. Yeah. One of the midwater fish.
It's another cyclothone.
Yeah.
There's a group called the bristlemouth, and there are very large bristlemouths also, but the cyclothone mature at a very small size.
And I think you said earlier, Mike, isn't it correct, or the estimate is that they're the most abundant vertebrate on Earth? There we go.
That is correct. And there are a few species.
Oh, this is interesting.
Species, but, okay, that's something.
Yeah.
What do we got?
What? I'm getting kind of a mollusk vibe, but I'm really confused.
Yeah.
I wonder if it's a larvacean that's out of its house.
Ah, that's a possibility.
Google. Well, yeah.
Yeah, I think you're right, Mike.
The same thing.
Oh, that's great. It's great because we've been seeing these larvation houses, but that's the individual outside of its mucus house. And as I understand it, they're capable of making multiple houses in a single day, depending on how quickly those filtration units become clogged, which is a really quite impressive use of the carbon. There's there's another arrowworm. Here's something in the middle. Yep.
Dougal was saying that larvacean was called a hammerhead larvacean. First time he typed hammerhead, I thought he saw a shark somewhere.
So that's the house without a larvation in it.
We actually saw him swim out of the house.
Right,
Alan, I don't know if you were saying the same thing as Roland.
Yeah, I was just mentioning that we could see the larvaceans leaving the house. But they are difficult to collect because of that. When you go to collect them, often the larvacean animal runs away.
Huh. Oh, I hadn't realized that. Interesting. Here we go. Oh, it's something on the bottom. Nice. And there's a fish also on the bottom. This is weird doing this so close at the bottom. I think earlier Dougal suggested we take our eyes away from the bottom. Something just drifting across from left to right, just coming into center now. Another larvation house. There's lots of them. It's still in there, yep. Still larvation inside.
When we were diving with the mirrors in the Charlie Gibbs fracture zone, the dominant fauna in the benthic boundary layer was larvaceans, which was interesting because the dominant benthic fauna was sponges, and both larvaceans and sponges feed on very small particulate matter.
So they're competing, was a suggestion. There's something I think coming in center.
Utilizing the same resource, I guess.
Yeah. Yep, here's another larvation house. And I'm not seeing the larvation in there. Maybe. No. Well, this has a different structure at any rate. I think we're seeing the internal filters of a larvation house, is my guess.
Yeah, like Dougal mentioned in the chat room, those inner filters look very different depending on what angle you're looking at them from.
Yep.
Because they're basically bilobed, and if you look at them from the side, you just see one of the lobes, and it's real different than if you look at it straight on.
So Pilot identified this one.
That's
so
a benthic sea cucumber.
Yep. Benthic sea cucumber is sort of chilling out in the water column here. Go ahead, Mike.
Yeah, they swim up into the water and then drift along and then settle back down to feed in a different area.
That's the head end. That's at the upper end.
Internal organ.
Yep. And you can see the oral feeding tentacles are what are sort of opening and closing at the top there. So the mouth is just in the center, so you can kind of follow the pharynx and the intestine back from there, and then it coils up, and that bright white material you're seeing are the sediments that it's been feeding on when it's settled on the bottom.
Dougal noticed that there's a small amphipod riding on the side of it, or like what I'm guessing is an amphipod.
So then shouldn't we call that thing a seahorse? Yeah. I don't think that's a larvation house. I think that might be something else. Copy that, Nav. Thank you. Oh, here's that larvation, the hammerhead larvation is what Dougal called it before. Not in its house. It's a nice view. You can see that tail is the lower part, very flat.
It's in the Fritillaridae.
Fritillaridae, it's the same sort of name as some of the butterflies. So what does that term mean? There's something, maybe a jelly, just coming across left to right, upper center. Yep. Oh, I see. Is that some kind of Nemerdian that we're seeing on the side? Well, when I first saw it, I thought I saw a dome coming below it. So is that an internal organ of something? Let's see. Okay.
Yeah, I don't know what that is.
Yeah. It's not moving at all. It's not giving us any clues. Yeah, it's hard for you to tell, but it's kind of tucked in really close to the ROV right now, so it's hard to get in there, and that's why you're getting that really hot lighting flash. Boy, it kind of looks molluscan, doesn't it? Oh, there we go. That must be a Nemerdian. Must be a ribbon worm.
Oh yeah. Yeah, I think you're right. It's a pelagic Nemerdian.
Yeah. Oh, that's very cool. Thank you for swimming. So there are some pelagic Nemerdians, I learned. Yeah, Dougal agrees. Good stuff.
Now turn and get a better look at it.
Yeah.
Probably not very well known, but also probably difficult to catch.
Yeah, John Norenberg has got an interest in these.
Very cool. Dougal gives a sort of a common name for it, the cow tongue worm. I don't know if he just made that up, but that's great. So the Nemerdians, that's a whole phylum of their own, ribbon worms, and we don't often see those on our dives. Most of the known species that I'm aware of are benthic, but there are some that stay up in the water column. That was a nice observation. I don't know on shore if anybody knows if that was a pelagic form or if that's just one that's swimming up from the bottom while it's young. I wouldn't.
No, I'm pretty sure that was pelagic.
Yeah, I wouldn't think that at least the benthic forms I'm familiar with would spend any time in the water. Yeah, go for it. It's another cyclothone, the midwater fish.
Starting to get an idea of why they're thought to be the most abundant vertebrate.
Yeah, there's a reason we're seeing so many of them.
Right.
I believe the abundance estimates were in the quadrillion.
Wow. That's amazing. So this looks like another mysid, I believe, an opossum shrimp. Get some confirmation from shore. That's a nice view.
Yeah.
Yep. It's hard to tell there, but the mysids, the females, form these plates that come down from the thorax, which is that part of the body just behind the head, and they will brood their embryos for some period of time, and they will basically live hatch the embryos instead of having larvae that swim around on their own in the water column. Another Ketignath arrowworm up here. Oh, no, my bad. That was a midwater fish, the cyclothone again. Check out this white thing. This is an amphipod, maybe?
Yeah, I think it is. Dougal says it's an amphipod, too.
Yeah, it looked like, there were a limited number of pleopods, those swimming legs. Amphipods have 3 pairs of swimming legs, and the shrimp would have 5 pair. Number of other characteristics that are different as well. I think we have, we're just about getting to the 10-minute mark, 10 minutes left in this transect. I'm not sure what...
That
said that was a gammarid I'm sorry. Dougal said that was a gammarid amphipod, which is not pelagic. So that came up off of the bottom, probably. The pelagic group are the Hyperiids.
We're looking at another larvation house, and you can see the tail beating on the upper side of it, and the brighter gray is the inner filtration. You can just see around that inflated is the mucus house surrounding it. So I think from shoreside they're saying that anytime you're ready you can turn on the ctenophore beacon.
Yeah. So I was kicking myself for not saying collect that thing as soon as we saw it, what it was.
Well, it's kind of like what Mike said, we don't know what we're going to see. Once we see it, we want to get the good image. Then often we don't have time to collect it, but the next one we will. It's a tough game here.
Yeah.
Yeah, not like the benthic stuff that sticks around.
Yeah.
Wait for you to collect it.
Oh, there, upper left. I was hoping it was a cephalopod when I saw it at first, but oh, there we go. Yeah. All right. We got people much better qualified to talk about this than me. I hope that wasn't Alan hanging up in excitement.
Well, we've got a little Hydra Medusa there. Yeah, it says Arthropodema.
Select this.
If you can collect it, pilot it. Oh, it's already past it, yep.
Wow.
Good effort. Oh, he went in. I think he went in. I'm watching the canister. It was so small.
Yeah, really small.
Yeah, I mean, I'm sure I saw it go in the hose.
Like it went.
But we can't see it in the canister right now. Well, I'll go ahead and I'll hit the collect button, just in case.
Yeah, I think you're going to find it when you're done.
Yeah. Do you want to suggest a name for me, Alan, for the collection database?
It's Arthropodema.
Arctopodema, thank you.
Yeah, Arctopodema.
Arcta, okay.
With an A.
All right.
2 A's.
Arctopodema, got it. That was exciting.
Yeah, it's a really interesting Trachea Medusa.
Let's see here. Too close, too close.
Oleolus.
That's what I thought, yeah. Yeah, like a salp. A little barrel-shaped salp. I can't tell if there's a Hyperion inside of it or if that was just its guts.
Dougal suggests that this is Paradoliopsis, perhaps. And I think what we were seeing inside was its gut.
Okay, thank you.
Not too long ago, I was annotating midwater stuff with Isabel, a Holling scholar who's on here. And we took a picture of that, something that looked very similar to that, and sent it off to Dougal to say, "What is this?" And then he sent the paper back where he had described some of it.
I've modified Stream 3's quad split to have more cameras, including the sample jar.
Thank you. The sample. I don't know if you heard that on shore, but the sample jar, the canisters for the suction are now as part of the quad screen, so you could look with us. Looks like we got about 5 minutes, just under 5 minutes left in this transect.
Yes, we did hear it, by the way, which is good.
Thank you, Mike.
Another mycid.
Another lopogastrid. Dougal comments that finally he got someone to read one of his papers. That's how it feels sometimes.
Yep. On the left of center, white. And the larvation house again. Yep. No, just to the upper left, though, there's something pink. Thanks. Huh. Yeah. I don't know what it is, but it's very small, and it's descending.
Maybe a little baby feeding nest.
Yep.
Come on, Tina 4. Show up.
I'm not sure what I'm looking at. Just a bigger chunk of marine snow, I think. 1 minute left in the transect, folks. Right of center. It's another larvation house. Yep, you can see the tail beating nicely. Okay, thank you. And right again, far right, lower quadrant. Oh, it disappeared. Oh, there it is. Good eye. That's a Keatignath, I'm pretty sure. Yeah, those are the gonads. So The body is divided into 3 regions, and so that central region, the longer white bars are the ovaries, and the end is the testes. And that's the end of our transect, 45 minutes. So that's the end of our benthic boundary layer transect, and we're now going to move up to 1,200 meters depth. We're currently at 1,817 meters depth. And so that's going to take roughly 20-25 minutes. And in that time, we're going to have a planning call for tomorrow's dive. And so we're going to temporarily switch out that camera 3 and put up the maps. And our expedition coordinator, Derek, has joined us, and he's going to guide us through this process. Can't hear you. I can't hear you on the OKEX line.
Thank you, Scott. Roland, if we could put up MB Proc 2 left.
Copy. The phone lines have been taken off the broadcast. Science, which computer was that?
MB Proc 2 left. Great. Okay, so on stream 3, you should see a Fledermaus planning scene that Shannon walked through this morning. So I'll just give an overview of where we're going tomorrow and a couple of the dive options that we have planned out before us. So our dive today is out here in this vicinity where my cursor is now. And overnight we'll be doing some mapping of some of this unmapped territory to the south here. And that'll give us time to transit over, do some dive options close to the main axis of the Mid-Atlantic Ridge. So,
Derek, can you point where we did dive 1? I think it's just to the north of,
yeah,
right there.
Basically this topographic high on this seamount here with the red peaks. So yeah, that was dive 1. So the next dive tomorrow will be in the close vicinity to that. And again, the reason for that is we had planned a bunch of things up north that we got kind of weathered out of. So we've been staying down here just below that low pressure system and getting what we can, exploring this region, which is fairly poorly unexplored. I did want to also point out these very small red dots for today's discussion. Those are actually magnitude 4+ earthquakes that have happened in the last month or so. Is that right, Ashton?
Yeah, that's correct.
Okay, so we have a couple of those highlighted here. There's some kind of up along the rift to the north, and then there's a few down on these orange dots, which are our draft dive track options that we'll be discussing now. So as you can see, there's one kind of up here, there's one in the middle, and there's one in the southern region. So I think I'll just give a quick overview of those 3 options, and then we can dig into whichever one kind of looks the most promising for people, and I'll ask Ashton to discuss a couple of them. The first one I wanted to point out is the furthest to the northeast here. And in the multi-beam data, we had just noticed there was this sort of Pancake Volcano feature that we see commonly in the Pacific in certain areas, but pretty unusual to see in this terrain. We've seen a few of them, but not many that have this morphology. So that would be an interesting dive target potentially. And that's located at about 3,240 meters depth. And what you see there, the sides on this feature have about 250 meters of relief from the surrounding seafloor. And one of the thoughts we had too is we could run over this with a multi-beam overnight and take a look at the backscatter and see if there's low backscatter signature on the top here, it could indicate that it's heavily sedimented. It might be less desirable.
I just got back. I had to take a nature break. We haven't started, right? Good. Thank you. Too bad that wasn't part of the transect.
Scott, while we're setting up, I could give a little bit of background about why we're doing this depth.
Yeah, sure. Go for it. That'd be really nice. We're pretty close to getting ready to go, but go for it, Mike.
Okay. Well, we're still in the bathypelagic, so we're deeper than 1,000 meters deep, and 1,000 meters is about as deep as sunlight can penetrate on a sunny day at noon. So we're in the part of the ocean where there is no sunlight penetration. And the bathypelagic is the largest ecosystem on the planet, but it's also the least studied ecosystem on the planet. So anything we see here is likely to be something that's really interesting and possibly important to know more about. And there's not going to be a whole lot in this transect, but we really need to do more work in the bathy pelagic because it's so poorly known, even though it's the biggest part of living space on our planet. And I think I've said it before on some of these dives, that if an alien came and just randomly sampled our planet, an alien species wanted to know what lived on Earth, and they took a random sample, they would take a sample of the bathy pelagic, and they would probably get a jellyfish. So
that's right.
We're looking at what might be sampled.
That was great, Mike. Thank you.
Uh-huh.
Lucky alien.
He's really lucky he'll get a squid.
Dougal Lindsay has offered an interesting observation as well, and he said that this depth, this bathypelagic depth that Mike was talking about, is also the target depth for the release of the return plumes during deep sea mining. So when they're churning up stuff in the bottom, this is where it would be ejected, I guess. So it's also important to know. Is this a Solmissus coming up ahead? What's jelly?
Yeah, Dougal's requested collection of Solmissus if we see one.
Yeah, I don't know that we're able to do that right now. They're still setting up.
Okay. I think his original request asked that we count how many tentacles it's got before we've collected, but I don't think we can do that.
If we count fast.
If it goes by that fast, it's going to be tough.
Copy that, Nav. Everybody is itching and ready, so we will do that. So on shore, we're counting down about 35 seconds. We will formally begin the transect at 13:32 time at transect 1,200 meters depth. I can hear you. Thank you. All right, 5, 4, 3, 2, 1, begin transect. There we go, siphonophore right off the bat. Or is it? Actually, this looks like an egg.
That's a radiolarian.
A radiolarian? No, I was going to say it was an egg mass.
Maybe.
I was going to say it's a salt chain.
All right.
Good start.
It's really small, and I guess it's really close.
Google suggests that was Colodaria.
Colodaria, I'm not sure what group is that in.
So that's a radiolarian. So whoever voted radiolarian is the closest.
Thank you.
Ooh. Nice. Okay. I think that's Genura. That's a Narcomedusa. Nice. Yeah, that is Genura Grimaldi. You can see that it has very small tentacles, really close to the bottom there. So you can see the large tentacles that you can recognize it as a Narcomedusa because the tentacles come out on the top of the bell. And then You see the large tentacles there, and if we get a little close up, we'll be able to see really tiny tentacles along the bottom.
And there's a shrimp in the background.
Yeah, there they are. Gorgeous.
Good stuff.
Oh, that footage is nice.
Thank you, pilot. There's the shrimp Mike was referring to. There's two, looks like a couple of Keating, yeah, and I think I'm seeing a couple of Keating-nass. We're just spinning the ROV around to the proper heading. Oh, that's the same Genura we're just looking at. All right, we're back on our heading. To the right of center, coming into center, it's going down. No, it's moving too fast. Don't go for it. It's really hard to get the. There's a jelly on the right, far right. There, it's center now.
Oh.
Got right up in our face. There we go. This is, I think, the Genura again. Yeah, we can check it. I think it's an arrowworm, a Keating-nass, I think, but let's have a quick look. Yep. Pretty sure that's what that was. Stand by on that, please. This is a ctenophore. Is this the same ctenophore? We'd really like to see this. If
I think it might be.
Yeah.
Can we collect?
Yeah, I think it went over the top of us. Boy, they're small. There's there's some kind of ring that just went by. Looked like one of those candy bracelets.
We're actually seeing a lot more stuff than I expected to at this depth.
So onshore, we have the ability to take 3 more water samples, the Niskin. So let me know if you want to do it at this depth. We'll save one for the deep scattering layer, but we have 2 others to decide. So let me know what depths you want to do that at. You can just put that in the chat.
I think one at this depth. Okay.
Yeah, go ahead, Alan. Follow up.
I was just going to say, I think it would be a good choice at this depth to correspond with the transect.
Okay.
And
I agree with you, Alan. I think it'd be great to get it in this because we don't really sample the Southeast project that often.
All right, that's good. That's 2 votes. Thank you.
I'll make it 3 votes.
Okay. So co-pilot's watch lead too. Regarding the Niskin, I believe it was you that was asking me? Yeah, so we would like one at this depth, so I don't know if operationally that's good at the end of the transect or during, you just whenever. Okay, thank you.
Taking a Niskin sample here. Reset.
That's another arrowworm, keating gnat.
Fire. 1339. D2 depth 1200.
All right, we got our eDNA sample. Thank you, everybody.
So does anyone know if there are several of the orange species, or is it something that's variable within a species? Just curious if anyone knows.
I think there's a genus that's reddish like that. It's in the bathypelagic, but there's a few species within that genus.
Now we got a suggested, I think.
A suggested shrimp, yeah. You can nicely see the elbows in the antennae. Those antennae have environmental sensors spread along them, and as they swim along, because the antennae are held out away from the body, but the long part of the antenna is parallel to the body, it functions sort of like the lateral line on a fish, but it gives them early warning because it's set so far out from the body.
Four for the water, yep. Looks like a mucus string, I think. There's a little copepod. Yep. There he goes.
That's really what the aliens would catch.
Yeah. It's another keating gnat. So, what I'm hearing, we're seeing at least, 2 or 3 different species of these arrowworms, these keating gnats, and I'm just basing that on the appearance of the gonads and what they look like.
Mm-hmm.
Was that the Genura again? Or is this different?
I believe that's right.
Okay. That's the one we imaged earlier.
You looking up.
Another copepod at the top of the screen. I think that's Cyclothone, one of the midwater fish. Yep. That's okay. We know what that is. That string that's going across the top. Yeah. Oh. Is that a little siphonophore, maybe? Oh, and let's check out this little pancake that's floating. There we go. That's a new one.
Yeah, that's a little true jelly, Schizozoan. I think that's
It's very interesting, Alan, what a flat sub-umbrella that is.
Yeah, I think Jugel agrees. It's a baby or juvenile Paralia.
Oh, really? So that really large sort of brown one that we saw earlier on in the dive, so this would be a juvenile. Oh, that's cool.
Mm-hmm.
Paralia. It's the genus name. There's another shrimp swimming on the lower right. Like maybe another mysid on the possum shrimp. Cyclothony fish just went through the bottom of the screen. Not sure what's coming across the center now. A little pink thing. And no, sorry, it was the other direction. It faked us out. Yeah, it faked us out. Let's have a look top right. Well, yeah, what's going on there? Oh, wow. So this is, look at this larvation host. It's huge. You can see the larvation, I think, in the center.
Oh.
Is that what that is?
Oh, no.
No, it's siphonophore. Oh, yeah, you're right.
All of its tentacles deployed.
Yep, it was kind of in a little bit of a circle, so it looked like a ball, but it was a chain that was kind of wrapped. Lensia siphonophore.
I just find that Lensia. A lot of times these siphonophores will sit out there. They deploy all their feeding polyps out.
This is another siphonophore, Alan.
And then waiting for food.
That one that had the
another siphonophore. It might be the same.
Yeah, let's check it out.
Almost looks like a loose tentacle.
That's what I was going to say. Disembodied tentacle or just a mucus string again. Okay, we can move on. There's another jelly at the top center. It's got a pointed dome on this one.
Yeah, here we have a halicreatid. This is another Trachea Medusae family. I'm sure that Google will be typing in the genus and species. I think it might be Botronema. Yeah, Botronema is what he says.
There's
so two really common Trachea Medusae families are the Halicreatidae and the Rhopalomatidae, we've been mentioning several times. The Halicreatids, you can tell them apart usually because they have wider radial canals and a really circular mouth, as opposed to the Rhopalomatids have sort of a mouth of lips. Or mucus there? Or is that a...
I don't know.
Yeah.
could focus, yeah, what's at the lower end there? Yeah. Huh. I don't know. I don't know if it's mucus. Yeah, thanks. I don't know if that's mucus or it's, you know, it almost looks like a ctenophore tentacle ripped off a ctenophore, but not sure.
Yeah. Floating piece of string.
Yep. String is the formal name that we have. This looks more interesting, though. This again looks, you know, I said egg sac, but I think someone before identified this as a kind of rhizarian or a
Colodarian. Yeah.
Yeah, Colodaria. So did I hear before that Colodaria, that's a kind of radiolarian or phaedarian?
A kind of radiolarian.
Huh, wow.
Distinct from the phaedarian.
Crazy, so a large single-celled organism. Swinging around again to get the current coming into us.
Genura again? The Narcomedusa?
What's the dark thing to its lower left? It's sort of, it's going with it. Or maybe you were turning. It was a dark blob on its lower left. Okay.
Mm-hmm.
Another one of the cyclothony midwater fish.
It always amazes me how similar the cyclothony and the Keating-gnats.
Oh, wait, pilot, pull out on your zoom. Upper left, I want to look. I think I saw a little dinner plate jelly, and we're looking to collect one. Yeah, is that? Let me check if that's the Solmissus, or is that the Solmissus that we're looking for? Or is no? I don't.
Does not look like Solmissus
yeah,
to me.
I don't think it is, but we'll have a look at it anyway. Tentacles are too short. Yep.
We have Halocreus minimum. This might be the spiky one.
Okay.
Yeah.
Thanks. I think that other string was just a mucus string, so I wanted to have a close look at that. There's something doing backflips just right of center now, and it's low between your toes. Okay, it's down below us, okay. You have a better sense of the orientation than I do. I get lost.
Easy.
Is the camera pointing down, though, right now? Oh, I see. Copy that. Thank you. Okay. Let me know when you're ready for operations. Copy.
So recap, we've got 3 water samples taken so far today. We've taken 1 biologic sample from all species and all those still pretty good. That's the last one. 1.6.
They've lost focus control on their camera. They've lost focus control on their camera.
Mike, just note your microphone's open. Mike's mic.
I'll turn it off.
There's that brown blob I saw before. Lower left, lower right. Yeah. Yeah, that's okay. I understand. Earlier I saw, we're just resetting the power on the cameras, folks, so don't worry about what's going on the screen here. Earlier I saw Dougal had put something in the chat. He said, was that Vampirocrosoda? And I'm wondering if that's what that dark blob that I saw, if that's what he was referring to. I'd like to see anything that has Vampira as part of its name. Oh, I guess we have the... I don't know which camera's on. Is it that one? Yeah.
No, we get to see the we get to see the control room. It's really nice to be in the control room right now.
All the cameras and computers we're responsible for, not the cameras. We're not touching the cameras. You want to smile, Arvind?
That's a special place to be.
Well, here's not the ctenophore we were looking for before, but I know this is a lobate ctenophore. That'll be good to practice the focus on.
I think that's Bathycyroe.
Bathycyroe. Great. So just for you folks on shore, we're troubleshooting the camera so you can't see the main feed. So on camera 1, you are seeing the control room. But as soon as we get this focus issue worked out, we'll switch back to that main ROV camera. So you can stick on camera 1, or if you prefer, go to camera 2. You can see these D2 in a wider view. So just to remind you, if you're just joining us on shore, we're just troubleshooting an issue with the camera, so stand by and we'll get that camera one feedback to you shortly, hopefully. All right, folks on shore, so just an update here. It sounds like what we're going to do is we're going to switch to another camera that is not the main camera, not the main Zeus. We'll go to HD2. And so you may notice that the angle is somewhat different. This camera is mounted on the upper bar of the ROV. Great, thank you. Yep. Okay. So this is a HD2. It's mounted higher on the front forward side of the ROV, so we're going to be looking down a little bit more, but otherwise we should be able to focus and zoom as normal. So shortly we'll get back to our transect. Yep, Nav, go ahead. That checks with my notes. Correct. Copy that, thank you. It should be jar one, although we did rotate the canisters to have a look. You saw it? Oh, great. Nice. Why is it when we're not doing a transect that all these things start swimming through the frame slowly? Pilot, watch C2. Can I just clarify that we are still in a holding pattern? We have not resumed the transect? Thank you. Great, thanks. Copy that, thank you. Looks like a little Keating-Nath, I think, arrow worm. Dougal Lindsay, who's done a lot of this midwater work, has said that he's never seen this many Genura on a dive before. That's these little sort of top hat jellyfish that we're seeing. is interesting.
I think that's a Coelodendrid Phaedarian.
Thank you. So I didn't, Alan, I didn't get the first part of the Phaedarian, so I'm not sure if you'd have to spell it in the chat for me.
Yeah, Dougal just typed it in, Coelodendridae.
Got it. Coelodendridae, thank you. Interesting, those tentacle-like extensions of the cell. I assume that, is it like binding up particulates from the water column and then phagocytizing those?
That's my understanding.
It's an interesting little jelly mimic. Yep. Uh-oh. I think this is the collection target. Is it, Dougal, is this what we're looking for? Maybe, I don't know. I don't know. Maybe not. Hang on.
Halocreus minimum.
Okay. Halocreus.
Halocreus minimum.
All right. Not a collection target. Thanks.
Jellyfish that has spikes. Yeah.
Alan, did you say 6 arms?
It's a really...
So they're branching arms?
No, I didn't say branching arms. No, these are individual tentacles that we can see, but what we see also on the top is the spikes on this jellyfish. So this is an unusual sort of armored morphology.
Oh, very cool.
That's fantastic. And there you can see the big open mouth. This is typical of the Halitriatidae family. Dougal says that those are probably used to break out of the stomach of gelatinous zooplankton like Solmissus when it gets eaten.
That'd be crazy.
What do we have there? This could be our Solmissus.
I'm standing by. Bingo. Yes, this is the collection target.
Yes, that's the one. Please.
We're going for it. Stand by. Yep, that's Paralia up there. Bingo! In the canister. Very nicely done. Folks on shore, it was pretty amazing. Keep in mind that the pilots are using a camera that they don't usually use, so it looks fully intact and it's in the canister. Very nice. I got so excited I forgot to push the collect button.
I had a little delay, so I got to know that it was successful after it happened.
That was really nice. So, Dougal Lindsay in Japan had earlier offered his kingdom for that collection, and so this is excellent. Our pilot, Lars Murphy, I've been waiting to use this nickname, the Count Basso, and I think that now applies since he has a kingdom waiting for him in Japan.
Way to go, Lars.
Arvind, if you check the chat, Dougal's got some instructions for how to preserve this. I believe that was a bonus. That was not on the transect.
That's great. So the transect has not restarted?
That is correct.
Thank you.
Got another suggested shrimp here. Again, you can see the bends in the antenna.
The reason that collection is so important is that Dougal and colleagues, they're working on straightening out the systematics of the Solmissus, different Solmissus species. It's been a very challenging group to work with so far, and getting some really good data. This will be a nice extra data point for that analysis.
Go ahead, Nev.
I, for one, have
copy that.
Published some sequence data for different Solmissus in the past that have the wrong names on them. So we've been busy fixing those in GenBank too.
Copy that. Thank you. Okay, so folks on shore, we have resumed the transect. We have 23 minutes remaining. And center left, not sure, no, it's under the light bar, never mind. I gotta remember I'm looking down a bit now. Yep, that's good. I believe this is the A. Genura jelly that we've been seeing fairly frequently, but I'll let the more learned Midwater folks tell me. Maybe not.
You got it, Scott.
Yeah, did I? Okay.
It is the same Genura Grimaldi.
I was starting to doubt myself. I couldn't remember if the other one was this pigmented. That's good, Pilot. Thank you.
Yeah, they're pretty dark red. Looks like it has a couple tentacles nibbled off.
Yeah. What do you think nibbles them, Alan? I know.
Good question. I don't know.
I've seen, in the Brusca textbook, figures of some hydrozoans munching on others, but this looks like... I guess it's Keatingnath. I guess so. I was thrown off a little bit by that bulb at the top.
Yeah, I agree.
Adrian Copeland says Keatingnath, narrow arm. Thanks.
Very, very fat.
Yeah.
Chunky.
Yeah, Dougal Black Gonads is wondering. That was the thing. The color just threw me off, especially at the posterior end, or what I was interpreting as posterior. Looks like another. I guess we're getting in position right now, so we'll stand by. Okay. Thank you, Pilot. So we're ready to begin our zooms again, if anybody spots anything. Is that something, or is that something on the screen? Something on the screen. Just seeing a lot of marine snow and mucus so far. Caitlin, are you able to look back at that last? Okay, tell me when it's good. I can stand by, don't worry. The last Keatingnath that we thought we were looking at was kind of fat and had the black bulb above it. It was about 1037. That's another cyclothony midwater fish. We've seen lots of those. We're okay on that. There, they are, that thing. Yep. Yep. I don't know that it ever gets a great focus. Okay, sorry. This looks like it's a siphonophore, or a new one for the day. So this is definitely worth imaging.
Very spiky.
Yeah, I don't know what I'm looking at. It's nothing like I've ever looked at.
Voglia.
Voglia. Huh.
Yeah, sometimes these little the little pointy bits, they're very hard, and they'll wash up on shore and be a really strange sight on the beach.
So are those pointy bits like the nectophores, the swimming bells? Or are they shields?
I think they are the swimming bells.
And presumably that long white dotted thing is feeding tentacle?
Yeah, well, that's its stem, and then off of the stem are the different types of zooids, including the feeding.
Oh, I see. Okay. So it's a long stem and then lots of very small zooids. That's probably the white that we're seeing.
Yeah, exactly. So the stem's pulled all the way in.
Very good. Thank you, pilot. And for Mike Vecchione and Shore, I'm looking at the video as rewound, that last thing that we were calling a Keating-Nath, and I think Mike's right. I think I see a head with a mouth and eyes. It looks like, to me, my inexperienced fisheries biology, it looks like a little leptocephalus larva head down. Copepod flying by. Copy that. You know, that last siphonophore, I'm more used to seeing gelatinous organisms with sort of smooth, rounded edges and not these pointy edges. Upper right. We're getting
now
there.
we've seen the spiky jellyfish as well as the pointy siphonophore.
Yep, and here it is again.
Yeah, there's a dipheid siphonophore.
Sorry, when I said here it is again, I meant what we were seeing on that benthic boundary layer transect. Rocket ship siphonophore, says Mike. Chunifes multidentata.
Chunifes again.
Maybe upper left, just left of center, and it's drifting left a little bit. Now it's kind of in the center left. I don't know how far or close that is. Yeah, it's so little. I can't. It almost looks like a pteropod or something. Okay, that was closer than I thought. It's interesting because I thought it's interesting that resolved into a slender little thing. I thought it was just a ball of detritus. On the upper right, I think that white thing drifting up towards the top of the screen looked like it was wiggling to me. Yep. Well, oh yeah, it did break in two. I thought it was S. Okay, we'll call that schmutz. There's a jelly on the upper left. That's good focus.
Oh, yeah.
Copy. Okay, so we're 10 degrees further down into the water. Gives us a little bit more time to see things as they come up towards us. I think I see something. No, it's below us. Yep. Can't tell if this is holding something. It's got yellow at the end of its tentacles, and I don't know if that it's captured something or.
Oh, it looks like.
Got out of our range, unfortunately. Copy that. Yep, looks like a large larvation house. It's definitely what it is. And I don't see anything in it, which is why yeah, no, there's a little guy right in the center there. It'll build another one. I'm going to guess that I'm going to be told this is the Paralia.
Yeah, for sure.
Okay. It's a good image, though. We were able to put a name on it.
That's really nice. It's relatively close relative to the common moon jellyfish.
Okay, we're ready to call things out again, folks. There's a little arrow worm going off the lower right. Top center, I see something. I'm getting to learn these ones. Looks like the A. Genura again. So still. Alan, does anyone know, is this the way it hunts? Does it just like lie in motion like this until something contacts the tentacles?
Yeah, quite a number of jellies. This is their sort of MO, is that they'll be sitting out there, more or less sit and wait predators, and then, only really swim hard when they're trying to reposition or escape predators. Oh, this is great video.
Yeah, I guess that's a good strategy for conserving the energy down here where food is hard to come by.
Mm-hmm. This really contrasts, though, with the Paralia, which is more of a cruising jellyfish with lots of fine tentacles. So the Paralia is more of a sort of standard filter feeder, cruising through the water and picking up small things on its tentacles.
That was a really nice view. Nav's watch lead 2. Can you tell me how much time is left in the transect? Copy that, thank you. About 6 minutes ago in the transect at 1,200 meters. Possibly coming in to the center on the center left. Very little guy. It's just looking like mucus to me now. Thank you.
Dougal just made an interesting observation, or shared an interesting observation about this Genura Grimaldi eye that we're seeing so much of. He says they encounter, one or two a dive in Japan, in Japanese waters, out in the Pacific. But this is the type locality for the species, the North Atlantic.
So we're in their wheelhouse. That was a little arrowworm, Ketignath, that we were zooming in on. Lower center, something slender and long, right in the middle. Yep, that's it. Yeah, just another mucus string. Oh well. Yep, upper left. Yep, looks like it's kind of pinkish on the left side. All right, that's what I'm talking about. That's not mucus, at least.
Ooh.
What am I looking at? Something with an intestine, I think.
I'm confused.
Yeah.
Geobius is a question mark from Mike Vecchione, which is a pelagic worm, annelid.
Huh. Fascinating. Copy that. So this will be our last observations for a couple of minutes here. We're just getting the pilot set up. A polychaete worm with no segments. Well, that really throws things off. Yeah, I just Googled that, and I do think that looks a lot like what it was. It's like a copepod there, the way it's moving right in the center. Oh, no, it's not. It's a ketognath. There's another one. Yeah, I think it's another ketognath, but we might as well check it out. Yeah. Is this a siphonophore in the foreground, though? Yeah. 1 minute in our transect.
Yeah, I think that is a siphonophore. I think maybe a praed.
Praed? Okay.
And Google confirms that actually in the subfamily Praenae.
Yep. Classic marine snow. Mucus and organics. All right, so that's the end of our 1,200 meter transect. I think we saw some pretty interesting things there. So now we're going to rise up in the water column another 300 meters to 900 meters, and then we'll resume our next transect. And I am going to use this opportunity to grab my lunch.